Hi everyone, my name is Adasa Sarabia. In this video, I'm going to explain and discuss the urea cycle. Urea cycle is a biochemical pathway that converts ammonia present in our body to urea. This happens because ammonia cannot be dissolved in water easily and is more toxic than urea. While urea can be easily dissolved in water and less toxic compound than ammonia. Basically, we can excrete urea in our urine and not ammonia. But before that, we need to understand where ammonia comes from and how it is transported to the liver where urea cycle happens. We all know that proteins is the most abundant organic compound which constitute an average of 10 to 12 kilograms of dry body weight of an adult. It performs a variety of functions in our body. Almost all biological processes, proteins are required. Proteins such as enzymes, hormones, clotting factors, and receptors. Half of the body proteins are found in the skeleton and connected tissues, while the other half is present in our cells. 1 to 2% of the total body proteins are turned over each day, especially into muscle proteins. Proteolysis is the breakdown of the proteins into individual amino acids, which are nitrogen-containing compounds, making the proteins as major source of nitrogenous waste, which are highly toxic, and to think that millions of proteins are broken down in our body every single day. When we look at the structure of the amino acids, it consists of nitrogen in the form of an amino group. So, as proteolysis happens, the nitrogenous wastes produced in the processes must be taken into the liver because liver will convert it into a less toxic water-soluble compound called urea. The nitrogen in the amino group cannot be released in our body directly because ammonium ions are highly toxic in our body. Thus, a process exists to transport the source of nitrogenous waste without converting it into its ionic form or ammonia because it can reach our brains that could result to worst-case scenarios. There are two prior steps that lead to the urea cycle. These steps are necessary in order to transport the nitrogenous waste to the liver without releasing it freely in our blood. First step is the transamination, or the transfer of amino group. The amino group in an amino acids which contains a nitrogen is taken from the amino acids and is transferred into some other molecule. That molecule can be mostly an alanine. And upon receiving the amino group, it is converted into glutamate. The glutamate can freely circulate the blood, carrying the excess nitrogenous waste without releasing it in our circulation, so that it can be directly transferred into the liver without releasing into the blood. When glutamate enters the liver, it undergoes the second step, which is known as the oxidative deamination, or the removal of amino group. The glutamate is the only amino acid that undergoes oxidative deamination to a significant extent to liberate ammonia for urea synthesis. The liberated ammonia in the mitochondria of the liver by oxidative deamination is then used to synthesize urea by a biochemical cycle known as urea cycle. Urea are less toxic than free ammonia and is water-soluble compound, which can be excreted in the kidneys in the form of urine. The urea cycle that was first discovered by Hans Krebs and Kurt Henslet in 1932 is known as the Krebs-Henslet cycle or the ornithine cycle, and later described into more detail by Ratner and Cohen. The urea cycle is a 5-step cycle with 5 distinct enzymes. The first 2 enzymes are present in the mitochondria, while the other are present in the cytosol. Carboxyl phosphate is the product of one molecule of ammonia and carbon dioxide. The reaction involves the carboxyl phosphate synthetase 1 or CPS1 located in the mitochondria and uses 2 molecules of ATP or adenosine triphosphate. The phosphate in the carboxyl phosphate is derived from 2 ATP only. ATP is converted into ADP. The whole reaction happens in the mitochondria only. The N-acetylglutamate or NAG is needed for the activation of the CPS1. Another reaction happens in the mitochondria also. The carboxyl phosphate combines with one molecule of ornithine, leading to the formation of citrulline. The ornithine, which is an amino acid, only functions in the urea cycle. It picks up the ammonia in the mitochondria by attaching the carboxyl phosphate to itself, forming citrulline. With the catalysis by ornithine transcarbamylase, also known as OTC, the carboxyl phosphate group is donated to ornithine and releases a phosphate group. The citrulline leaves the mitochondria, then enters the cytoplasm for the urea cycle. A condensation reaction occurs between the carbonyl group of citrulline and the amino group of aspartate, forming argininosuccinate. This reaction is ATP dependent and is catalyzed by argininosuccinate synthetase. This reaction, the ATP is converted into AMP, an inorganic phosphate group gets released. The argininosuccinate is broken down by argininosuccinate lyase to form arginine and fumarate. The arginine is then cleaved by the enzyme arginase to form urea and ornithine. One molecule of water is also used in this reaction. The ornithine is then transported back to the mitochondria to begin the urea cycle again. The urea passes into the blood, and since it's water soluble, it can easily get excreted with urine in the kidney. To sum it up, there are 2 prior processes that happen before the urea cycle. The 2 processes are transamination and oxidative deamination, which are important to prepare the amino group for urea cycle. It is needed to happen so that the nitrogenous waste cannot go into the circulation, which is toxic in the body. The urea cycle involves 5 steps and 5 distinct enzymes that help break down and remove nitrogen from the body. In the mitochondria, one molecule of ammonia is combined with carbon dioxide, forming carboxyl phosphate. Then the carboxyl phosphate is added to ornithine to form citrulline. The citrulline then leaves the mitochondria, then enters the cytoplasm. In the cytoplasm, the carbonyl group is condensed with the amino group of aspartate to form argininosuccinate, catalyzed by the argininosuccinate synthetase. The argininosuccinate is cleaved by argininosuccinate lyase, forming arginine and fumarate. The arginine is then broken down by the enzyme arginase to form urea and ornithine. And the ornithine will be back in the mitochondria to begin again the urea cycle. The urea will pass the blood to the kidney for excretion together with the urine, since urea is water soluble. Before I end this video, I would like to thank Med Simplified for the information about the urea cycle. That would be all. Thank you.